Okay, ladies and gentlemen, in our last video, we discussed the foundational ideas of Mendelian genetics. So for today's video, what we want to do is discuss what we call the exceptions to Mendel's rules. But what they really are layers of details that we add to the basic ideas we know. For instance, when it comes to plants, we learned from Mendel that you can either have a tall plant or a short plant, and there was actually nothing in between. And so when genes were passed on, you either had heterozygotes that were tall, or you had homozygous dominant that was tall, homozygous recessive that was short, and that created ratios like we're familiar with our 3-to-1 when it comes to things like a heterozygous and a heterozygous cross. But if you were to take a look at something like human height, what you'll notice is that there is not an either or. You are not short or tall. We have actual cross measurements across all levels. I mean, you could be so tall you don't fit on the height chart, or you could be finding yourself somewhere near the bottom. And we have every possible height that spans in between. And so that's just one example of the things that we want to cover when we're going to talk about what we call the exceptions to Mendel's rules. So let's talk about our first exception, which is known as incomplete dominance. So typically when we talk about Mendel's rules, what we're discussing is this idea that you have a dominant and a recessive. So let's go with flower color in plants. If you have purple flowers, all right, you could be homozygous dominant, or you could be homozygous recessive, or I'm sorry, heterozygous. If you have white flowers, you are homozygous recessive. Now, this is flower color in peas. But that's not the only way that flower color can be done. Over here, you have a type of flower known as a snapdragon. And what you see is that snapdragons can be red, they can be white, or they can be purple. And what's interesting here is, when it comes to the peas, it was purple or white, and your genotype only could give you one of those colors. Whereas here, with the snapdragons, it's red, white, or purple. And so there has to be a way for your genotype to give you those colors. And so the basic understanding here for incomplete dominance is that the heterozygous genotype is going to give you your own phenotype. So before, this heterozygous genotype right here would give you the dominant phenotype. But here, and you can see this in the Punnett square to the left, the heterozygous genotype is actually giving you a unique phenotype. In this case, it's pink. The reason this happens is because on a biochemical level, the secretion of proteins and enzymes that is leading to the color in the flowers, up here in the pea plants, the purple is all that shows up biochemically. Whereas down here, what's happening is the colors are mixing so that the red is showing up and the white is showing up, and it's actually creating that blend that, you know, forever people thought that's actually how all traits were passed on. And it turns out it's not all, so a blend is not all, but it is only one possibility. One of the things that you will note most when it comes to incomplete dominance is that there are always 3 phenotypes, and that the genotype ratio is almost always equal to the phenotype ratio. which, as we've learned in our work so far, is not the case with simple dominance or the basics of Mendel's rules. Okay, so now we want to get into exception 2, which is known as codominance. Now, codominance is a little bit tricky to explain, so you're going to have to bear with us as we give you an example here. So the first thing that you have to understand about codominance is that it means that every allele is present in equal amounts. So when it comes to incomplete dominance, the dominant allele is still sort of, the most present. It's still the one that is, sort of being altered a little bit by the recessive allele. Whereas in codominance, every allele is present in equal amounts, all right? There is no blending, okay? So there's no blending. It's not like incomplete dominance. So let's talk about our example. Our example is generally used as human blood type. So human blood type actually has a couple of possibilities. You could have A blood. You could have B blood. Or you could have AB blood. Or you could have O blood. So for human blood type, we have 4 possibilities here, and basically what we have are 4 phenotypes. So let's talk a little bit about what these phenotypes actually are. So when it comes to human blood, okay, you actually have red blood cells. And your red blood cells, like any blood cell, will have proteins on the surface. Okay, so there's going to be proteins on the surface here. And hopefully you remember, okay, from our previous discussion of the cell membrane, that these proteins are actually in the membrane. Okay, so they're actually like in the membrane. They are most likely peripheral. So they're actually proteins that don't extend all the way through, but are on the outside, although some are transmembrane. But the key here is that these blood proteins, all right, they are used to help identify the cell. We mentioned that one of the most important things when doing cell membrane structures and the way that your immune system works is that your body needs to be able to tell the difference between self and non-self. Okay, hopefully you remember that. And so what happens is your body has to have some way to identify what cells belong to it. And it has to have ways to identify which cells do not. This is actually how you fight off invaders. So these blood types here are a way for your body to self-identify. Okay? Now, what happens... No, we're not going to do that yet. We need this. Okay. So what happens with blood type is, if this is your red blood cell, okay, what you might have on your red blood cell is proteins that look like this. Obviously, they don't look exactly like this. This is simply a representation, okay? And this might be what the A blood protein looks like. And then let's say that you have a blood cell that looks like this, okay, and it might have on it proteins that look like this. Let's actually fix this one. Oops. No, there's the eraser, there's the eraser. Okay, let's fix this. Okay, so this would be the B blood protein. All right, so when we're talking about the phenotypes, what we're saying is that you either have the A protein or the B protein. Now, if you were AB blood, what this means is that you actually have both proteins. So you might have the square and the circle, and the square and the circle, and the square and the circle. And that's why it's codominance, because you have both equally. All right? That's the key here. It's both. So this, of course, begs the question, what about O blood? Okay? So with O blood, you would actually have a red blood cell that does not have the A protein and does not have the B protein. Okay? So none of these proteins. And so what this does is you can actually have A blood that is A plus A, because remember you need two alleles, or it could be A plus O. So you could have A and O because then you're expressing the A blood and the nothing. You could have B blood that's B plus B, or you could have B blood that's B plus the O allele. Okay. Now down here, you have AB blood. The only way to get this is to have one copy of the A allele and one copy of the B allele. And then down here you have O blood where none of these proteins are present. And the only way to have that is to have one copy of the O allele and one copy of the O allele. So what this does is this creates a very specific Punnett square. We do this to represent A blood. We do this to represent B blood. And we do that to represent O blood. So what you would do is, let's say I have someone who is A blood, and they're heterozygous with O. And I have someone who has B blood, and they're heterozygous with O. When I mix them in the Punnett square, I would do IB, IA, and that person would have AB blood. Down here I would do IA and I. Over here it would be IB and I, and down here it would be II. So what this does as a phenotype is this person is O blood, this person is B blood, this person is AB, and this person is A. Most important things to remember here. Every allele is present in equal amounts, and it's not a blending, it's a presence of all. So even though O blood means you don't have those proteins, you could see right here, if you have the B blood protein, you're expressing the B protein. If you have the I protein, you're expressing the I protein. Okay, I think that that's actually all we're going to cover for the exceptions in this presentation. We'll go through some of the others in our next one.